Hi, welcome to this MicroGEM webinar. This is the first of a series of webinars that will explain how we can simplify your sample preparation workflows. You'll be producing high-quality DNA and RNA with good yields, less chance of error, and less chance of cross-contamination. In this webinar, we will tell you a little about MicroGEM, who we are, and how we reinvented nucleic acid extraction. To achieve this, we've cut through the dogma and gone to extremes, literally. Oh, and if you are wondering about the GEM in MicroGEM, it stands for genes, enzymes, and microbes. MicroGEM began life in the 1980s as Pacific Enzymes Limited. We were a company working out of two New Zealand universities, and our strategy was to discover new organisms and enzymes in extreme environments. The goal was to identify robust enzymes for industrial applications. New Zealand was an obvious place to do this work. On the Pacific Ring of Fire, the country has geysers, hot pools, and volcanoes that were a ready source of new, extremely thermophilic bacteria and archaea. At the other end of the scale, New Zealand has good access to Antarctica, the coldest, driest, windiest place on Earth. From the huts and artefacts of the great Antarctic explorers like Scott and Shackleton and Amundsen, the team accumulated a wealth of cultures and knowledge. From the extraordinary McMurdo dry valleys to the boxes of food and supplies in Scott and Shackleton's huts, 110-year-old carcasses and the meat locker, and a mummified seal on the floor, all unique culture mediums for extraordinary microorganisms that have had over a century to grow. These expeditions of discovery gave the resource on which MicroGEM was founded, an expansive curated culture collection of fungi, bacteria, and archaea. Organisms found from -10 degrees C to +95 degrees C, and in addition to these, we accumulated environmental samples ready for metagenomic sequencing. Today's company is run by an executive team with over 175 years of experience in business, plant sciences, microfluidics, instrumentation, and molecular genetics. Our company is spread across the globe with a head office in the UK, microfluidics and engineering in the USA, manufacture in China, and biology in New Zealand. So how is MicroGEM different from other life sciences providers? MicroGEM extraction kits and instruments use cocktails of enzymes that work at different temperatures. This means that we can create a sequence of coordinated reactions by simply changing the temperature, and that, of course, is easily done using a thermal cycler, something we all have in the lab. You don't need expensive robots and instruments, just the biochemistry that comes in the MicroGEM kits. Using this innovative approach, we've been able to cut through the dogma. We've thrown away the recipe books for molecular biology that we've slavishly followed for 25 years. The dogma around how we do things in the lab is ingrained in all of us, and so we use complex methods for no other reason than we have always used complex methods. Even when companies introduce sophisticated automation, the same old chemistries are inside, making the instruments large, complex, and expensive. It sounds a little unfair to suggest that brilliant scientific minds stick to rigid rules, but handling DNA is tricky, and you often can't even see your sample. So if you have a method that works, why change it? The reason for change is obvious if you think about it. When you are doing 1 or 2 extractions a month, a little inefficiency doesn't matter. But when you are doing hundreds or even thousands of samples, time and money tell you to change. MicroGEM has reinvented the biochemistry. We have removed hurdles that we have doggedly lived with. Let's for a moment look at the current ways we extract nucleic acids. Whether you are using beads or columns, almost all methods start with proteinase K. Now, a good side to proteinase K is that it works in strong solutions of ionic detergents like SDS. The bad side is that it actually needs those detergents to make it effective. And once you've put SDS into your lysate, you've created a whole lot of problems for yourself. You have to get it out. The proteinase K is okay. You can kill it by simply heating your tube to 65 degrees C, but the SDS has to be removed. Otherwise, it will inhibit every downstream reaction you choose to do, whether that's PCR, qPCR, WGS, or STRs. The easiest way to demonstrate what this limitation does to your workflow is with the oldest solvent extraction method. You follow the proteinase K step with a phenol extraction. This is to remove the protein and the SDS. You've now successfully got rid of the SDS, but you've put phenol in your extract, which is as bad as SDS. So you first extract with a 50-50 mix of phenol and chloroform to help things along, and then follow it with a chloroform extraction. The phenol has gone now, but now you've got chloroform in there, and that is worse than phenol. So you add sodium acetate and precipitate with alcohol. That gets rid of the chloroform, but now you have acetate in your DNA. So you rinse it with 80% ethanol, which gets rid of the acetate, but you still have ethanol in your DNA, so you have to dry it down. If you look at every step in the method, each is to remove something you put in there in the previous step. The steps have nothing to do with the starting sample. Now you may think that columns and beads are better, but they're still doing just the same, going through a series of bindings and washes and elutions, all tedious and prone to mistakes and all causing yield loss. Why? Because you put detergent in your extract. You may ask, what about the other components you are getting rid of? Well, the main ones are debris, which you can spin out if it's a nuisance, and hydrolyzed protein, something you will probably add to your subsequent reactions in the form of BSA. So why are you so anxious to get rid of it? So what can we do to improve things? Proteinase K is rapidly inactivated above 55 degrees C. Despite what some methods tell you, it's stone cold dead at 65. The problem is that protein is much more easily hydrolyzed when it is denatured. That's what the SDS does. But heat works just as well, and so MicroGEM found a thermophilic proteinase that works optimally at 75 degrees. Now you have your denaturing conditions without a detergent. At MicroGEM, we went through our culture collection and tested a large panel of enzymes. We had a wish list. We wanted a proteinase that was thermophilic and optimally active at around 75 degrees, but it shouldn't be too stable because we needed to kill it by raising the temperature a little more. It should also have a broad specificity and, importantly, work in buffers compatible with PCR and other downstream reactions. After screening our collection, we found a proteinase in a bacterium found in a hydrothermal vent high on the slopes of Mount Erebus in Antarctica. Mount Erebus is a 12,500 foot active volcano which, like Kilauea in Hawaii, has a liquid lava lake in its crater. We named the bacterium EA1, Erebus Antarctica Isolate 1. So now we had a proteinase that could do everything that proteinase K could do, but without a detergent and in simple buffers compatible with PCR. We could lyse the cells, digest the proteins, and use the DNA directly without bothering with all the purification steps. A second advantage to using EA1 proteinase is that normally it's impossible to mix other enzymes with proteinases. For example, how do you mix lysozyme with proteinase K without the proteinase destroying it? The answer is you can't. There are plenty of published methods that try to mix the two, but when our team followed the progress of the reaction, we found the lysozyme was destroyed in the first few seconds, so it served no purpose. That's not the case with our enzyme. EA1 is inactive below 45 degrees. It's effectively deep frozen, so you can put the two enzymes together in the pot, incubate at 37 degrees, and let the lysozyme do its work. Once the lysozyme has done its job, you raise the temperature to 75 degrees C. The EA1 becomes active, ruptures the cells, removes the nucleosomes, kills any nucleases present, including tough RNases like RNase A, and removes the lysozyme. It needs to be removed because lysozyme is very inhibitory to Taq DNA polymerase. A last raise to 95 degrees and the EA1 self-digests, so it can no longer harm the Taq. So, how does this help? Well, the obvious advantages are reducing time and effort, which reduces costs, and also removing complexity, which reduces error rates and yield loss. But also, you can use this strategy when it is impossible to carry out multi-step extractions, as is the case with single-cell work, sub-microliter PCR, and digital PCR. You can't add easily new reagents once your reaction has started. With the MicroGEM method, all you do is add the reagents and heat step your reaction through the stages. There is, of course, a little more to our products than just EA1 proteinase. For 15 years, we have worked on proprietary formulations and strategies. Our two families of kits are PrepGEM for the life sciences and ForensicGEM for forensic biology. These kits come with everything you need: enzymes, buffers and additives, easy to follow instructions. We are also aware that scientists around the world are extracting DNA from 1,000,001 different things. And for many, there are no off-the-shelf instructions. But please, if you are working on some strange creature that we've never tried before, send us an email and we'll try to help you get things going. And if we can't help you, we'll be honest with you. MicroGEM hasn't stopped at reagent kits. We took things a step further with the invention of the PDQEX, which, as you have probably guessed, stands for Pretty Damned Quick Extraction. The PDQEX adds to the biochemistry of temperature-controlled extraction by providing automated purification through a polymer column. This column sits at the bottom of a disposable cartridge. We still use temperature to control the reaction, so the PDQEX is very simple. It has only one moving part, the fan to cool the electronics, hence it's low cost and robust and can be powered by a transformer or a battery. The PDQEX works by heating the extraction reagents in steps, either a single 75 degree, or if there are other enzymes in the mixture like lysozyme, it will use a lower temperature step first, just as we do with the reagent kits. After the 75 step, the temperature is ramped again. At 80 degrees, a heat burstable valve ruptures and the inner tube of the cartridge, which is made of thermoresponsive plastic, shrinks. The extract is squeezed through the purification column and into a collection tube. The polymer removes polyphenolic and carbohydrate inhibitors and, importantly, the EA1 proteinase. Because we don't need an EA1 heat kill step, the DNA does not reach above 80 degrees C and so stays double-stranded. Furthermore, the purification step removes typical inhibitors from soil, stool and plants. So the DNA is ideal for constructing libraries and next-generation sequencing. What's more, because there are no pipetting steps, the extracted DNA is minimally sheared, and so you get long read lengths with PacBio or Minion sequencing. The ease and speed of the PDQEX are demonstrated with side-by-side comparisons of competing procedures. These are the recommended methods for extracting bacterial DNA for NGS using columns, magnetic beads, and MicroGEM. There is no competition. And remember, fewer steps means fewer mistakes, less time, higher throughput, lower cost, great yields, better read lengths, and less plastic waste. The million-dollar question is, does it work? These results show a few examples. The left is a human STR from a sperm stain extracted with the forensic GEM sperm kit. The center image is a qPCR of the human BRCA1 gene, extracted from a buccal swab using the PrepGEM universal kit. And on the right are comparative plots showing Illumina sequences of Myothermus ruber when DNA was extracted using the PDQEX or QIAGEN. With the plant DNA, we can isolate PCR-ready DNA for PCR and qPCR in less than 15 minutes. Samples are collected in the field using our collection tool and storage cards, and punches from the cards go directly into the PDQEX. There is no need for liquid nitrogen or grinding, and no tedious purification steps. The method, known as PhytoGEM, has been validated on a wide range of plants, pathogens, and sample types for PCR and qPCR. We will talk more about the MicroGEM kits and instruments in later webinars. From this one, we hope you have gained a little insight into how we can help you, either if you are doing 1 or 2 extracts or many thousands, and we'll work closely with you to help you optimize your particular application. In the meantime, you can get more information from our website, microgembio.com, and don't forget to email us at info@microgembio.com if you have any questions. Thanks for taking your time to listen to us, and we hope to hear from you soon.